Hello everyone, today I would like to present about diversity of marine phototrophs. My name is Eli Jesina binti Abdul Rahman and I am from course Marine Science. Firstly, what is phototroph? Phototroph is transformation energy of solar radiation into chemical bond energy. And what is primary producer? Primary producer is organism that acquire their energy from sunlight and material from non-living sources. They also can produce ATP using sunlight energy. In the middle of 1970, two microalgae was detected as primary producer. But why? This is because they can easily speculate on phytoplankton sampling approach and the method for phytoplankton detection. Symbiotic dinoflagellae in coral at tropical reef made them as primary producer at oligotrophic ocean. What's meant by oligotrophic ocean? Oligotrophic ocean contain low level of nutrient and almost free from biogenic activity. During this year also, phytoplankton research using DNA detection and flow cytometry shows that haptophytes including Phototrophs are one of the major constituents of primary producer. Late in 1970, the development of observation technique by fluorescent microscopic and collection knowledge from electron microscopic observation exposed a unicellular cyanobacteria Synechococcus as a primary producer at tropical and subtropical region. It has a very tiny cell and called as picocyanobacteria. 1979. AAPB was derived from non-sulfur purple bacteria, which is the anaerobic and oxygenic photosynthetic bacteria in alpha proteobacteria. It contains bacterial chlorophyll A, and as reported, phototrophic growth of some AAPB was faster than those growing heterotrophically in the dark. Next, anaerobic and oxygenic photosynthetic bacteria, which is using bacterial chlorophyll to perform photosynthesis without oxygen evolution. These chlorophylls are detected in algae and plants. Alright, before go to the next slide, what is different between oxygenic photosynthesis and anoxygenic photosynthesis? Oxygenic photosynthesis happens when H2O serves as electron donor to replace the reaction center and form oxygen as byproduct, while anoxygenic photosynthesis occurs when others reduced compounds serve as electron donor and oxygen is not generated. In late 1980, a development technique of HPLC for pigment analysis and cell detection using flow cytometry detect a picocyanobacteria Synechococcus. This cyanobacterium name is Prochlorococcus and it contains divinate chlorophyll A. This cyanobacterium using divinate chlorophyll A to perform oxygenic photosynthesis and can grow under deep water. Chlorococcus using divinate chlorophyll A to perform oxygenic photosynthesis and can grow under the deep sea water column where blue lights are dominant. This is because divinate chlorophyll can absorb blue lights more efficiently than monovinate chlorophyll. In 1996, a cyanobacterium Archaeochloris marina was detected, which is this cyanobacterium can form oxygenic photosynthesis using chlorophyll D. What is chlorophyll D? Chlorophyll D is redshifted chlorophyll which can absorb far-red light for 30 nanometer longer wavelength than chlorophyll A. This cyanobacterium using chlorophyll D not only for light harvesting antenna but also for chlorophyll in reaction center. In 2000, there are widely distribution of aerobic and oxygenic photosynthetic bacteria AAPB which is contain bacterial chlorophylls and also proteohydrosin that containing bacteria. Both of them are photoheterotroph, which is without carbon fixation and has been never recognized as primary producer. In 2011, a new candidate phylum was detected using metagenomic analysis targeting plastic DNA. It was given name as Rapemonas and very close to haptophytes, but it did not belong to any known phyla phylogenetically. Metagenomic analysis in 2007 detect a phytoplankton which did not belong to any known phyla of algae. It was given name as Picovilliphas and distributed widely in coastal area of Europe and North Atlantic. In this year, another redshifted chlorophyll was reported from a cyanobacterium which is isolated from a microbial made on stromatolite. What is stromatolite? Stromatolite is layered sedimentary formation which is created by blue-green algae. Development technique of metagenome and flow cytometry were published which shows that significant amount of picohaptophytes including many undescribed species. Picohaptophytes form 500% of picophytoplankton biomass which contribute them to primary production in marine environment. As conclusion, the new discoveries and improvement of new technology shows that phototrophs and primary producers are almost the same. But all of this did not stop here because there are many microorganisms still need to be explored in this world. Thank you for watching my presentation. Bye.